The use of Y chromosome short tandem repeats loci in a case study by Adan Jose Esquivel Jr. What is Y-STR loci and mtDNA analysis? Y chromosome short tandem repeat loci system has the capability to determine the number of male contributors in a male-female mixture by allowing to isolate individual male DNA profile. It does this by focusing on the detection of male-specific genetic markers and the non-recombining section of the Y chromosome to establish a patrilineage. While mitochondrial DNA analysis is a commonly utilized method to examine genetic information within the mitochondria of human cells, found in forensic samples that are either degraded, decomposed, or of low DNA containing samples. mtDNA sequencing consists of 4 steps: 1. PCR amplification, 2. DNA sequencing reactions, 3. Separation using electrophores, and 4. Data collection and sequence analysis. How Y-STRs and mtDNA is utilized in forensic investigations. The Y-STRs is utilized in forensic investigations such as sexual assaults, paternity confirmations, and the identification of a missing person's remains. For mtDNA analysis is a crucial tool in forensic investigations involving mass fatality occurrences and identifying a missing person's remains as well. Utilizing Y-STR analysis of highly degraded DNA fragments from over 70-year-old human skeletal remains. The goal of the study, conducted by Key Laboratory of Evidence Identification in universities of Shandong Province and Shandong University of Political Science and Law, is to determine DNA extracted from the teeth of skeletal remains over 70 years old from a group of missing persons and to possibly identify a patrilineal relationship to either family 1 or 2. This study consisted of 6 skeletal remains of missing persons and 5 reference samples from potential biological relatives with the utilization of the Y-STR method for ancient DNA extraction. The Y-STR results generated would prove or disprove the alleged family members if they are from the same paternal line. Thus, Y-STR analysis is the best approach for determining potential kinship. Sample preparation. The DNA extracted from teeth is of higher quality than the DNA extracted from bones. Before beginning the process of DNA extraction, you must first remove any surfacing contaminants prior to drilling. The 6 samples of human teeth are first bathed in 5% Na hypochlorite for 5 minutes and rinsed with distilled water, followed with being soaked in absolute ethanol for 5 minutes and transferred to a clean disposable urinalysis cup to air dry overnight. Lastly, before drilling the teeth into powder, all teeth were exposed to UV light for 30 minutes all around. DNA extraction and purification. To maximize the DNA yield, the combination of large-scale silica-based extraction method with complete demineralization was implemented in this scenario, beginning with drilling 50 milligrams of powder from teeth and incubated for 24 hours at 37 Celsius in 1 milliliter of extraction buffer consisting of 0.45 moles over liters ethylene diamine tetroxylic acid EDTA mixed with 0.25 milligrams over milliliters of protein kinase with 0.05% of tween 20. Afterwards, the transferring of the supernatant into a 13 milliliter of binding buffer containing quintidiene hydrochloride to be purified with the silica-based minolute spin columns, and the DNA is eluted in 50 microliters of elution buffer. DNA quantification, amplification, and DNA profiling. The 3 commercial kits that were used for generating the Y-STR results are 1. Yellow Platinum PCR amplification kit consisting of 38 Y-STR locies and 3 indel locies. 2. GoldenEye DNA Y plus PCR amplification kit consisting of 41 Y-STR locies and 3 Y indel locies. 3. FastDirect DNA 44Y PCR amplification kit consisted of 44 Y-STRs and 3 Y indels, along including the core Y-STR loci database provided by the Scientific Working Group on DNA Analysis Methods and European Minimal Haplotypes. DNA separation, detection, and data analysis. The amplified DNA products are separated based on size and detected by the ABI 3500 genetic analyzer with a POP4 polymer and monitored by standard ejection parameters. The raw data is then retrieved and analyzed by Gene Mapper IDX software v1.5. An allelic ladder included at least once per injection on the 96 well plate. Calculation of likelihood ratios. In order to confirm the alleged relationship between the missing person and families, the likelihood ratio needs to be calculated with the patrilineal relationship versus the non-relationship of male pairs. HO is the hypothesis that the ancestor and offspring are related, and H1 is the hypothesis that the ancestor and offspring are unrelated. Y-STR results. The Y-STR haplotypes required from family 1b2 and family 1b3 were identical to each other, and the alleles in the consensus profile of the missing person number 7 at the 27 and 28 Y-STR loci. However, a possible single-step mutation could have occurred at the DYS437 locus, with an allelic variant 15 being detected in an individual of family 1. As for Y-STR, haplotypes obtained from family 2b1 through b3 are consistent and identical to the missing person number 18 at the 22 Y-STR loci, revealing it is 19,425 times more likely that Y-STR results from both missing person number 18 and the alleged relatives are paternally related as opposed to unrelated, while Y-STR haplotypes from missing person number 6, number 11, number 17, and number 19 aren't consistent with samples from either family 1 or 2. Explore why traditional STR analysis wasn't helpful in this forensic case. The use of Y-STR analysis over traditional STR analysis is a challenge of DNA extraction and profiling of skeletal remains, due to how often the state of remains is found degraded and/or offers low DNA quantities. Examples of these type of forensic scenarios include war conflicts, natural disasters, airline crashes, fires, and mass casualty scenarios. Compared to traditional STR analysis, autosomal STR markers provide highly accurate results for analysis of kinship, but not of distant relatives. Thus, uniparental markers are much more useful due to being more informative for this type of forensic analysis study. Conclusion. In conclusion, the Y-STR method in DNA genotyping is highly effective in DNA extraction and profiling on old skeletal remains, although with instances of allele dropouts were observed in all samples with a few drop-ins in some, with the likelihood ratio used as confirmation to the Y-STR results of the alleged relationships between the missing persons and their relatives with great probabilities, emphasizing that this study supports the efficiency in the utilization of Y-STR loci in DNA extraction from old skeletal remains. These are my references, and have a great day.